In this video, I will discuss ribose sugar and its function in nucleic acids. The most important sugar from the standpoint of genetics is the simple sugar ribose. Ribose C5H10O5 serves as the sugar backbone for ribonucleic acid RNA. Deoxy-ribose C5H10O4 is derived from ribose by removing one oxygen atom from the hydroxyl group at the 2' carbon atom. Deoxy-ribose serves as the sugar backbone for deoxy-ribonucleic acid DNA. Ribose and deoxy-ribose are the backbone sugars to which phosphates and a nucleobase are attached to form the nucleotides that DNA and RNA are built from. The DNA nucleobases are adenine A, thymine T, cytosine C, and guanine G. RNA molecules are built from the same nucleobases except uracil U is substituted for thymine T. When deoxy-ribose is bonded to the nucleobases, adenine A, thymine T, cytosine C, and guanine G, the result are the DNA nucleosides deoxyadenosine, deoxythymidine, deoxycytidine, and deoxyguanosine. When ribose is bonded to the nucleobases, adenine A, uracil U, cytosine C, and guanine G, the result are the RNA nucleosides adenosine, uridine, cytidine, and guanosine. The addition of one phosphate to any of these nucleosides would result in a DNA or RNA nucleotide that could then be used to build a DNA or RNA molecule. ATP, the energy source for many biologic functions, is formed by adding 3 phosphates to an adenosine nucleoside to form adenosine triphosphate, ATP. Ribose comes in 4 different forms and several pucker variations. All ribose has the same formula, C5H10O5. The 4 different forms are alpha-D-ribopyranose, beta-D-ribofuranose, linear D-ribose, and linear L-ribose. Alpha-D-ribopyranose is a hexagonal ring of 5 carbons and 1 oxygen with 4 hydroxyl groups OH. Beta-D-ribopyranose has the same hexagonal ring, except the hydroxyl OH group at the 1' carbon is up rather than down. Alpha-D-ribofuranose is a pentagonal ring of 4 carbons and 1 oxygen, and 3 hydroxyl groups OH and an aldehyde CH2OH at the 4' carbon. Beta-D-ribofuranose has the same pentagonal ring, except the hydroxyl OH group at the 1' carbon is up rather than down. The nucleotides for RNA are generally formed from beta-D-ribofuranose. There are also 2 linear forms of ribose. Linear D-ribose is chiral right, or has its hydroxyl groups on the right-hand side. Linear L-ribose is chiral left, or has its hydroxyl groups on the left-hand side. L-ribose is man-made and is not found in nature. In aqueous solutions of ribose at room temperature, 76% will be in the D-ribopyranose form, and 24% will be the D-ribofuranose form, and less than 0.1% will be in the linear D-ribose form. For the deoxy-ribose form used in DNA, start with a beta-D-ribofuranose and remove an oxygen atom from the hydroxyl OH group at the 2' carbon position. When the ribose is already part of a RNA nucleotide, removing the oxygen from the ribose is done by the enzyme ribonucleotide reductanase. The action of a ribonucleotide reductanase enzyme results in the conversion of ribonucleotide into a deoxy-ribonucleotide. The reason why the oxygen is removed is to make the ribose sugar less reactive. Oxygen is a highly reactive atom. Deoxy-ribose is more stable and less reactive than ribose. Deoxy-ribose is used in DNA nucleotides to make the DNA more stable. DNA must keep its structure stable and unchanging for long periods of time, so the stable, less reactive deoxy-ribose is used in DNA. RNA is a short-lived molecule that must interact and react with other RNA and protein molecules. The reactive ribose works for the short-lived, reactive RNA. The small structural difference between ribose and deoxy-ribose is shown in the figure. The figure clearly shows the missing oxygen at the 2' carbon location. The next figure shows a free deoxy-ribose as a beta-D ribofuranose with an oxygen missing from the 2' carbon position. Carbon atoms and hydrogen atoms are not shown but should be assumed by the viewer. A pucker variation is simply an atom in the ring bent out of the ring plane. The plane of the ribose ring is defined by the plane of the 2 carbons bond with the oxygen atom. Every carbon out of that plane contributes to the pucker of the ribose molecule. There are an infinite number of angles the other atoms can form with the ring plane. The pucker gives the ribose and the whole nucleotide flexibility in forming the spiral of RNA or the double helix of DNA. Puckers above the ring plane are called endo. Puckers above the ring plane are called exo. Some examples of pucker are pictured in the figures. Research scientist Gaspar Bonfalvi published his study in the article, "Why Ribose is the Sugar Component of Nucleic Acids?" in the journal DNA Cell Biology in March 2006, and reprinted on the NIH website I show. The pentose sugars, ribose and deoxy-ribose, are the only sugar components of nucleic acids. Bonfalvi modeled many other sugars as possible backbones for nucleic acids. He eliminated all other sugars as incompatible with nucleic acid structure. Based on his models, Bonfalvi concludes, quote, "Comparisons of sugar pucker conformations and configurations of pentoses indicate that ribose was not randomly selected, but the only choice. Since beta-D ribose fits best into the structure of physiological forms of nucleic acids. In other nucleotides containing arabinose, xylose, or lixose, the C2'OH and or the C3'OH are above the furanose ring, causing steric interference with the bulky base and the C5'OH group." Unquote. It's an important conclusion from Bonfalvi's work. We can state ribose and deoxy-ribose are the only sugars that work for building nucleic acids. If ribose did not exist, RNA and DNA could not exist. If ribose did not exist, life would not exist. If ribose is the only choice for nucleic acid and genetic coding, then ribose may be more important to life than glucose. There are no substitutes for DNA and RNA that we know of that can code genes. If there are other ways to code for life, all our experimentation has missed them. And any other way we imagine that life could be coded has, so far, failed to code reproducible processes necessary to sustain and reproduce life. In 2019, a team of scientists that included Yoshiro Furukawa and Bill Steigerwald claimed to have detected ribose in the Murchison meteorite. The Murchison meteorite fell on Murchison, Victoria, Australia on September 28, 1969. Observers said they saw the fireball at 10:58 in the morning. The meteor exploded in the air and scattered debris over a 5-square-mile area. One chunk of the meteorite weighed 15 pounds. The total weight of the debris recovered is over 220 pounds. The Murchison meteorite was found to contain a surprising amount of organic material, including amino acids, nucleobases, and sugars. The Murchison meteorite continues to be a source of active study. The Murchison meteorite is thought to be over 7 billion years old, which makes it older than the solar system. How all the organic material got inside Murchison meteorite, and how such complex compounds as amino acids, nucleobases, and sugars came to be synthesized is a hot topic of study and debate. Were they naturally synthesized? Are the organics evidence for extraterrestrial life? Is the Murchison meteorite a remnant of a planet with life that was blown to bits in an explosion 7 billion years ago? Do the organics present in the meteor lend credence to the panspermia theory that life was brought to Earth via meteors? Is this a limit on our ability to know the truth? Finally, I give the pathway by which cells synthesize ribose from glucose. I don't demonstrate the pathway to bore you. I give the synthesis pathway in hopes you will appreciate how complicated life is even in the transformation from the simple sugar glucose into the simpler sugar ribose. Some students of organic chemistry or biochemistry may find my demonstration useful. I refer professionals to the Ninja Nerd YouTube channel to get a fuller explanation of the details of ribose and ribonucleotide synthesis, at the link I show here. Pentose synthesis from glucose to ribose is not done in one step by a single transformation enzyme. Pentose synthesis takes 7 steps, 6 enzymes, 2 ATPs, 2 NADP+, and a water molecule. To transform glucose into D-ribose that could be used to build an RNA nucleotide, or reduced to deoxy-ribose to build a DNA nucleotide. In step 1 of pentose synthesis, the enzyme glucokinase uses ATP to add phosphate to glucose. The result is glucose 6-P. In step 2, the enzyme G6P dehydrogenase uses NADP+ to reduce glucose 6-P to 6-phosphogluconolactone. In step 3, the enzyme lactonase uses a water molecule to convert 6-gluconolactone into 6-phosphorgluconate. In step 4, the enzyme 6-PGDH uses NADP+ and the removal of a CO2 to convert 6-phosphorgluconate into ribulose 5-P. With the loss of a CO2, a 6-carbon molecule becomes a 5-carbon molecule. In step 5, the enzyme isomerase converts ribulose 5-P into ribose 5-P. In step 6, the enzyme RPP kinase uses an ATP into a MP reaction to add 2 phosphates to convert ribose 5-P and wrap the PRPP molecule formed into a 5-carbon pentose ring with 3 phosphates chained onto the 5' carbon. In step 7, the enzyme PR transferase strips 2 phosphates from PRPP to convert it into a ribose with 1 phosphate. The ribose phosphate is ready to become part of a nucleotide. How all these complex conversion steps would happen naturally, inside a meteor in outer space, without the benefit of enzymes to catalyze the reactions, God only knows. And that concludes my take on ribose. In my next series of videos, I will discuss how ribose is used in nucleic acid, and the general functions of the nucleic acids in DNA and RNA.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.